Okay. So, as promised, here's a quick video on the endocrine system. So we've been spending time in the nervous system. So remember that the nervous system impacts the neurons. So the divisions of the central nervous system, understanding that you've got electrical signals that are triggering the release of neurotransmitters and the gaps between neurons, and that's how that signaling is being processed. So your body essentially has two primary signaling systems. You have your nervous system and your endocrine system. And it's really important that you understand the difference between those two systems. So the nervous system is designed to work quickly. The neurotransmitters are designed to be in those gaps between your neurons a very short amount of time. But not all of the signals that get sent throughout your body need to move that quickly or should move that quickly. So the companion signaling system to the nervous system is the endocrine system. So the endocrine system uses hormones. Now, some hormones are super fast acting. Some of them are not. Some of them can stay in your bloodstream for hours at a time or days at a time. So again, we're going to do a fairly superficial job with the endocrine system because we need to understand as students of psychology just the basics of this. If you were to continue on, or if you wanted to get into neuroscience or something like that, then you would need additional classes and you need to go much deeper. So the major glands of the endocrine system. So you need to know these. There's some pictures in your book. So the pituitary gland, it's considered the master gland. It sends messenger hormones to control other endocrine glands. So it's directly responsible for the secretion of growth hormone, endorphins, and others. So the pituitary gland sends out the hormone signals to trigger other glands. So that's why it's called the master gland. Your thyroid gland, located in the neck, releases hormones that regulate growth, metabolism, and appetite. So again, hyperthyroid, hypothyroid. So hyper means too much activity. Hypo means not enough. There are certain disorders associated with irregular thyroid activity. So if you continue in nursing, you'll talk about those quite a bit, and probably touch on those in anatomy and physiology as well. Maybe patho. So your adrenal glands sit on top of your kidneys. They secrete hormones involved in stress response. So things like epinephrine, otherwise known as adrenaline, and norepinephrine. So again, your adrenal glands can be triggered by stress. So we mentioned neurotransmitters being triggered by stress. So again, you've got the autonomic nervous system, the sympathetic nervous system that will release the neurotransmitters, begin to activate your body's stress response. But you also need something that stays out for more than a fraction of a second. So then the activation through that can then trigger your adrenal glands to secrete hormones also involved in stress response. So sometimes that long-term chronic stress, what we see is not neurotransmitters are hanging out in your body forever, but that your adrenal glands are continuing to pump hormones out that are impacting other functions of your body, even if you're not still running from that bear. So your pancreas secretes hormones that regulate blood sugar levels, so insulin and glucagon. So again, in nursing, that's going to be something that you talk about a ton. So we're just mentioning that here. And linking it to the endocrine system. So you'll notice in this video, there's a hyperlink. I'm not going to punish you guys with that, but there is a Weird Al parody song done in a Beach Boys style, which may not mean anything to you guys, but might to your parents, about the role of the pancreas and hormones. It's kind of catchy. If you want to, it's honestly pretty good content too, to be honest with you. So if you want to hear it, you can do a YouTube search for Weird Al pancreas song. Okay, so then your gonads are general specific, so gender specific, sorry, and they release hormones that trigger secondary sexual characteristics. So this is the, your ovaries, release stuff. The secondary sexual characteristics are, you know, development of breasts, pubic hair, all that other kind of stuff. So all those functions are controlled by hormones. So this is where I kind of want you to live. These are the things that you need to know. I always have an essay question in the essay pool about the differences between hormones and neurotransmitters. So if you get it, make sure that you tell me what each one does and then directly make the comparison. Don't give me definitions about what they do and then make me make the comparison. So again, so if you get this essay question, make sure that you cover the key differences and their similarities. So differences in where they act, how fast they act, and how long they last would be good areas to concentrate on. And then the similar functions, remember that both hormones and neurotransmitters are involved in your body's messaging systems. Okay. So then here's another video link that I will drop in on our page. So again, this is the most heavily scientific chapter. So it'll get a little bit better for you guys. And if you guys have really enjoyed the science of this chapter, we got one more. And then not that there won't be science in the other chapters, but it will be more research based and not directly, you know, chemistry, physics, that kind of thing. Okay. So I'm going to go ahead and loop in some of our conversation about brains into the hormone video, just because I don't want to post 15 different videos for this week. So in psychology, it is expected that you know the different lobes of the brain, the different areas of the brain. Again, if you pursue psychology and neuroscience, you will talk about this in a lot more detail. So we're staying somewhat superficial, but you do need to at least know the lobes and their general functions. There's usually some type of matching question to cover this content in the test pool. So the frontal lobe is in charge of executive functions, thinking, planning, problem solving. It's at least partly responsible for emotional regulation, other things, behavioral control. So there's a lot of research that has kind of demonstrated that your frontal lobe is the last area of your brain to fully develop. So, you know, that has some interesting implications when we look and talk about things in developmental psychology. So if that is the last lobe of your brain to fully develop, a reasonable student would ask, well, what age does that fully develop? A lot of research is pointing to the idea that it fully develops around 25, 27 years of age. So for those of you guys that may be taking this class concurrently, you're still in high school, you still have some years to wait. For those of you that are returning to school after a brief stop out, you might be on the other side of that 25 to 27 window. So you might actually be better at those executive planning functions. So some of you guys that are a bit older and you are annoyed by younger people, their brains aren't done yet. So whether you're annoyed by people you work with or by your own kids, their brains aren't done yet. There's still hope. Okay, so other lobes. So the temporal lobe is linked to memory, understanding, and language. So if somebody has a stroke and they lose some language function, it is likely that we will see diminished activity in the temporal lobe. Occipital lobe is related to vision, controls vision. So there's a whole lobe of the brain dedicated to processing visual signals. If you think about that, that's just kind of a powerful thing to think about. So the parietal lobe, again, perception, making sense of the world, arithmetic, spelling. And then we've got the two cortexes up here, the motor cortex controlling movement and the sensory cortex controlling sensations. So the point that I want to make about this, and where some students can tend to get caught from time to time, is there are multiple areas of your brain that are active at any given time. So there's some thoughts. The idea that's been kicking around is you really only use 10% of your brain. That's not particularly well defined, and that's a little bit misleading. Your brain structure, we don't know just a ton about brain structures. The study of brains is relatively new. Up until just a few decades ago, it was very difficult to study certain things in living brains. And so if you review the different imaging techniques in your book, you'll get a sense of now the kinds of things that we can learn and study from living brains. But neuroscience as a whole is not a particularly old field. But one of the things that we've come to understand is we can do functional MRIs, and we can ask people questions and watch the different areas of their brain light up, is that these electrical signals that are, going through your brain that are involved in processing, there are multiple areas of your brain active at the same time. So it's not that a signal comes in through the occipital lobe and then you see it hop over to the temporal lobe, and then then it goes over to the motor cortex. Like, there are areas of the brain of activity throughout. One of the things that's been interesting, sometimes when students present on their psychological disorder, which again we'll talk about as the weeks progress, some students choose to include brain images. So like, one of the really interesting things is if you look at a brain image of somebody with ADHD and somebody that does not have that clinical diagnosis, the activity in their brains is different. Different areas are active. So again, super interesting stuff. I would spend the entire semester on this, and frankly, guys, you just don't need all that time on this, at least not at this level. Okay, so brain lobes. Know the brain lobes. So I included this diagram, and if you look at it, if you kind of match up where the visual cortex is between those two, so it kind of gives you an idea of orientation. Why I included this is that it shows you Wernicke's area and Broca's area and the primary auditory area. So if you look at kind of the features of that, and it's imperfect because it's not the same brain lobe diagram, but again, super terrible artist. If I was a better artist, I'd draw my own so they overlap perfectly, but here we are. So Broca's area and Wernicke's area cover different things in relation to speech. So make sure that you look for that in your textbook, because that explains some different types of aphasia and stuff. Again, if you do cover that topic for your research, you'll learn a little bit more about it. So anyway, again, so I made this point a little ahead of my slideshow presentation. Post-mortem exam reveals damage. So most damage is not pinpoint accurate. In your chapter 3, you will see a picture of Phineas Gage and a diagram of how they think the railroad spike shot through his head. So, if you were to watch a video of this or see 3D imaging, you would kind of get a sense for maybe the different lobes of the brains that were impacted and that kind of thing. But all you see post-mortem is damage. So when Phineas Gage died and we looked at his skull and we looked at his brain, we would go, oh, that's not right? Because we'd studied, brains of healthy people post-mortem, but it doesn't really tell you exactly what area of damage causes a particular type of loss of function. So early studying of neuroscience and brains were basically case studies. People like Phineas Gage that had damage, and then we kind of looked for changes in behavior or ability. So there was a patient by the name, I believe, of HM, again, for confidentiality purposes, we don't release real names, and suffered from very strong seizures. Very, very strong seizures. They didn't know what to do. And so what they did is they wound up severing the hemispheres of the brain, so the right and left brain. And they noticed that the seizure activity was greatly diminished. So in that way, it impacted quality of life. But again, because the brain is a cooperative entity with all of its parts, what they realized is that it impacted HM's ability to remember and recall new information. So unfortunately, early on, a lot of the case studies that we have were due to people, or a result of people, I guess I should say, that have had some type of brain damage studied. So lesioning and ablation, so animal studies. So some of you with queasy stomachs may not like this very much, but to actually intentionally lesion an animal brain and see what function they lose. So again, one of the criteria for doing studies is this research necessary? And, some institutional review boards approve that because animal models are the most ethical way to study it, because we can't just do that to people. So we had to wait quite a while for technology to get better. That a lot of it was post-mortem. There's all kinds of interesting treatments. In class, sometimes we watch like videos about deep brain stimulation, all kinds of stuff. But anyway, we had to wait for imaging techniques to get better. So if we were to continue on in this, you would learn about some of the different techniques and imaging techniques. For the purposes of this class, I'm going to let you go to your book to read that, because those things are pretty easy to understand out of your book. If you have questions as you're reviewing the imaging techniques, feel free to ask them. Again, email is usually better for me than other methods.